Welcome to AB Biofun with Dr. D. Let's warm our brains up and answer the following question. What is the smallest unit of evolution? And if you're watching this at home, please pause the video, answer the question before proceeding. So the smallest unit of evolution is the population. And in this series of tutorials, we're going to be talking about population, genetics, or how populations change over time, how they evolve. Okay, this is called microevolution because microevolution deals on evolution, small changes on a small scale, hence the word micro. So remember, we discussed the Grant's findings of the small ground finches in the Galapagos Islands after a change in the environment, the drought. So what changes occurred in the population of ground finches? Again, pause the video, answer the question, and I'm reminding you about the data by giving you a different version of the graph here. And hopefully this was your answer. The average beak depth increased in the finch population because birds with smaller beaks were less likely to survive and reproduce after the drought. The finch population changed, not individual members, but the population of this particular ground finch changed. Similarly, we completed the rock pocket mouse activity. And you see here a picture of the two main classes of fur color in southern Arizona. So what changes occurred in the population of the rock pocket mice? Again, pause the video and answer the question. The changes were that the fur color changed due to change of the ground color. The population of the mice that lived in a dark ground color where a lava flow had deposited had a different genetic makeup. So the population of mice evolved. Again, not an individual mouse, but the whole population. So the smallest unit of evolution is the population. And microevolution is defined as a change in allele frequencies in a population over generations. So there are three mechanisms that can cause allele frequency change. One being natural selection, which we explored with the finches and with the rock pocket mouse. Two, genetic drift, and three, gene flow. And we're going to explore each of these in this tutorial. Now, only natural selection, though, can consistently cause adaptive evolution. So genetic variation is what makes evolution possible. So the variation of heritable traits is a prerequisite for evolution by natural selection. Genetic variation can be measured at the whole gene level, that is gene variability, and can be quantified as the average percent of loci. Remember, loci is another term for genes that are heterozygous. Genetic variation can also be measured at the molecular level of DNA, which is nucleotide variability. Now, we've explored over the past year all the sources of genetic variation, but here we're going to explore them from a different point of view. Namely, we're going to talk about how new alleles arise and how new genes rise throughout evolution. So genetic variation can result when either new alleles are introduced or genes are introduced through either mutation, gene duplication, or other processes. So mutations can cause new genes, new alleles. Sexual reproduction cannot cause new genes or new alleles. It just shuffles. It creates genetic variation by shuffling, by arranging existing genes in new ways. So it's important to understand the difference between the sources of genetic variation. Sexual reproduction being just a way of shuffling and redistributing genes and alleles that are already present versus mutations and other mechanisms that actually can create new alleles and new genes. So how do we actually get new genes? Look at this information I'm providing you here. So E. coli, which is one of the most common bacteria we encounter every day, has about 4,400 genes. Yeast, Saccharomyces cerevisiae, the baker's yeast, has 6,600 genes. Now we have 20,000 genes, so we have a lot more genes than E. coli or yeast, which are both unicellular organisms. So how do we get this many genes? How do new genes arise? How do new alleles arise? So chromosomal rearrangements, duplications, translocations, we talked about these type of mutations. So they're not small mutations, they're large changes in large portions of DNA. And they actually contribute to what we call the evolution of genomes. They contribute to the fact that our common ancestor with E. coli and yeast had a lot less genes, but look at us now, we have 20,000 genes. We have different genes, or we have different alleles from yeast or E. coli, and they have different genes themselves, which are different from their last common ancestor. So this is called the evolution of genomes. New genes means new proteins means new functions. So remember that. So we're talking about evolution at the molecular level. So normally when we talk about evolution, we talk about new phenotypes, new features, new variations, new species, adaptations, but they're all based on the evolution of genomes. They're based on getting new genes, getting new alleles. So we're going to look at evolution at the molecular level. So how do new alleles form? Well, new alleles really arise by mutation. A mutation really is a change in the nucleotide sequence of an organism's DNA. So point mutations, we talk about point mutations, which are single nucleotide changes in the DNA sequence. And the mutations can be more than just a point mutation. It could be several nucleotides. So these mutations, insertions, deletions, or changes in the nucleotides can cause the formation of new alleles. Now, mutations that would change a phenotype dramatically, drastically, rarely will come for an advantage in a particular environment. So we don't really see these, but mutations that may cause small changes that could later be selected for, we may see in a population. Now, it's also important to understand that we can also get mutations in non-coding regions of DNA, either regulatory regions or other non-coding regions, and they can result in either neutral variations or they can actually cause differences in phenotype. Now, sometimes differences do not confer selective advantage or disadvantage. So this is how we get new alleles of existing genes. That means new versions. So new versions of existing genes. But how do we get new genes? How do we change the number of genes an organism has or the position of the gene? Now, mutations, again, these are caused, new genes are caused by mutations, but these mutations are on a much larger scale. They delete, they disrupt, they duplicate or rearrange many genes. So they're typically harmful, but not always. So you can duplicate a small portion of a chromosome and that may not be harmful. And if that duplication persists, because it's not harmful, over many generations, that duplication can actually accumulate mutations and thereby create new genes. So duplicated genes that are not harmful can accumulate mutations and take on new functions. Gene duplications are believed to play a major role in evolution. And I'm very briefly going to discuss now 3 major mechanisms of basically getting new genes and new alleles. 3 major mechanisms of genome evolutions. And they're not the only ones, but they're the major ones and the best understood. And in this example, I'm showing you the evolution of the globin genes, not the hemoglobin, but the globin genes. So the globin genes encode for the subunits of hemoglobin, fetal hemoglobin, myoglobin. Those are all the proteins that carry oxygen, responsible for the carrier proteins that carry oxygen. And due to analysis of DNA sequences, which I alluded to in our first lecture, we are actually able to trace the evolution of the genes. So what you see here is like an evolutionary tree, except instead of organism, here we have molecules. At the very top is the ancestral globin gene, which at some point in time was duplicated. And because it was duplicated, now technically what could happen is you could keep one copy unchanged and altered and then just mutate one. But sometimes both copies can accumulate mutations. And then these give rise to two different genes, and these different genes can now be duplicated and mutated further and so on. And ultimately, it results in 16 gene family members. These are called gene families because they code genes with similar but yet different functions and similar structure. So we have 16 of those on chromosome 16. Sorry, not 16, but we have, sorry, not 16. We have 11 of those on chromosome 16 and 6 of those on chromosome 11. So this many genes are the result of evolution at the molecular level, and they all we started with a single ancestral globin gene. Through duplication and mutation, we've acquired many more genes. Now, there's also something called exon shuffling, which is literally recombining the exons, which are the coding regions of gene, and creating brand new genes. And here the example is shown of the TPA gene, which is a shuffle, kind of a shuffle from a gene called the epidermal growth factor gene, the fibronectin gene, and a plasminogen gene. So you see how exons from these three genes were shuffled and assembled in a different way to create a whole different gene, okay? So finally, I keep talking about these jumping genes in class, and here they are again. We call them transposons or jumping genes. They're actually technically not jumping, because what happens is the transposon is actually copied, and that copy really jumps, and it can jump and insert itself in a new location, either on a different chromosome or a new location on the same chromosome. Now, these when these transposons jump, they can create mutations. They can create new alleles, or they can actually create even new genes, because when they jump, or rather when they're copied and then they go to a new location, they can also copy something other than the jumping gene. They can copy neighboring DNA and carry that neighboring DNA to a new location, and thereby contribute to that exon shuffling that I showed you earlier. So they are a driving force in exon shuffling. So all of these that I just told you, the duplications, the exon shuffling, the jumping genes, are really a type of mutations. They're the result of errors, and these errors are usually errors in meiosis and recombination. Remember the crossing over that occurs in meiosis. There's sometimes also errors in mitosis. So these are mistakes. These are errors, but these mistakes and errors can contribute to the evolution of genomes. They can contribute to the creation of new genes and new alleles. Now, some of these are dead end. Okay, they don't offer any kind of advantage. Some of these are detrimental, so the organism will not survive and reproduce, so we don't really see them. Then once we do see, however, our evidence that these things are happening, and the reason we see them is because they have conferred some kind of advantage. They have been selected for. All right, so now that we've generated variation, we're actually going to go back to how you create more genes and variation when we talk about how new species form. But let's talk about population genetics, or how can we tell if a population is evolving, if a population is changing. Now, genetic variation is required for a population to evolve, but it doesn't guarantee that it will. Just because you have genetic variation doesn't mean the population is evolving. So one or more factors that cause evolution must be at work for a population to evolve. And I would like to introduce you here to the concept of a gene pool. A gene pool consists of all the alleles for all the genes in a population, okay? So basically, every gene in the population and all the version of that gene comprise the gene pool of that population, okay? So allele frequency in a population, or population genetics. The frequency of an allele in a population can be calculated. It's very simple. Diploid organisms, the total number of alleles at a locus is the total number of individuals times 2, right? Because we have 2 copies of each gene. The total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual, right? Because the homozygous individual has 2 dominant alleles. Plus, you got to add the 1 allele from the heterozygous individuals, right? And the same logic would apply to recessive alleles. So recessive alleles, the number of recessive alleles is you got to count the number of recessive phenotypes, multiply by 2 because they have 2 recessive alleles, and then count the number of heterozygous individuals and add 1 to that because they have 1 copy of the recessive allele. Now, we also say that an allele for a particular locus is fixed if all the individuals in a population are homozygous for that same allele. So if, let's say we can have 2 or 3 or 4 or 5 or however many different versions you want for a given gene. And if all the individuals in the population are homozygous for one of these alleles, for one of these versions, then this allele is fixed. There's no other alleles in the population. Now, population genetics is a branch of evolutionary biology that deals with alleles of the whole population. So it's called genetics because it deals with alleles, it deals with genotypes and phenotypes, but it doesn't deal with genotypes and phenotypes at the level of a single mating pair. It deals with genotypes and phenotypes at the level of the whole population. So we call that population genetics. Okay, so gene pools and allele frequencies. So by convention, there are 2 alleles at a locus. If there are 2 alleles at a locus, because we could have multiple alleles for one gene, not just 2. But let's stick with 2 for simplicity's sake. So by convention, the 2 alleles are called P and Q. Usually P is the dominant and Q is the recessive allele. Okay, so if you have 2 alleles that are given for a given gene, then we say that the frequency of the dominant is called P and the frequency of the recessive allele is Q. Okay, and P plus Q equals 1. And right away, I want you to notice that when we say frequency, we don't mean percentage, we mean frequency. So that's a decimal number. Okay, so when you add them up, they should add up to 1. If it would be a percentage, they should add up to 100%, right? Because we only have 2 alleles. So let's consider the following examples. So consider a population of wildflowers that is incompletely dominant for color. So you have 320 red flowers. That is, red is the incomplete dominant allele. So they're homozygous dominant. You have 160 pink flowers, which are the heterozygous individuals. And you have 20 white flowers, which are the homozygous recessive alleles. So how do we calculate the number of copies for each allele? Well, we just count them, right? So we have 320 homozygous dominant. And since each homozygous dominant has 2 of the dominant alleles, we need to multiply this by 2. So that's 320 times 2. Then we need to add 160 dominant alleles from the heterozygous because the heterozygous individuals have 1 dominant allele. And when we do the calculation, it comes out to 800, all right? So how do we calculate now? The same way we can calculate the numbers, we're now calculating the numbers of the alleles. We can count the alleles of the recessive alleles. So we have 20 flowers, which are homozygous recessive. That's 20 times 2 is 40. And we have 160 heterozygous individuals, which carry 1 recessive allele. So that's 40 plus 160, that's 200, right? So we have 800 dominant alleles and we have 200 recessive alleles. And we did this by counting them, all right? So the total number of alleles in the population is 1,000. So how do we do the frequency of the dominant and the recessive allele? Simple. The dominant alleles, there was 800 of them, right? The total number of alleles is 1,000. So the frequency of the dominant allele is 0.8 because when you divide 800 by 1,000, you get 0.8. Or you can say it's 80%. But please be aware that if I'm asking you for frequency, you have to answer 0.8. If I'm asking you for percent, only then do you tell me 80%. 80% is not a frequency, it's the percent of alleles. So you need to be very precise in your answers, okay? Now, since we know that the frequencies have to add up to 1, we can immediately say that the frequency of the recessive allele, or Q, is 0.2, because 0.2 and 0.8 make up 1. Or it's 20%, because if you have 80% of dominant, 80% of the population has the dominant allele, sorry, if the dominant allele is 80% of the total gene pool, then the recessive must be 20, correct? Or we could have simply used, again, the formula saying, okay, we have 200 recessive alleles divided by 1,000, that would give me 0.2 or 20%. Okay, so let's talk now a little bit about the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is a mathematical model that is used to describe, to test whether a population is evolving or not. So the Hardy-Weinberg equilibrium is a hypothetical situation, and it serves as the null hypothesis. And we're going to get back to that, what that means. So if a population is not evolving, that will be our null hypothesis. Our null hypothesis is that there is no difference between what we see and what we expect if the population is not evolving. Okay. Now, a non-evolving population can exist only if we remove all of the agents of evolutionary change. And those agents are genetic drift, and we're going to talk about that later. Genetic drift, there's no genetic drift only if we're dealing with very large populations. No gene flow, which means no migration in or out of the population. No mutations, so there's no chemical changes to the DNA. There is random mating between the individuals of the population, so there's no sexual selection, and there's no natural selection at work, so all individuals have equal survival and reproductive success. Okay, and this is a hypothetical situation because it is very, very rarely met. Okay, so the Hardy-Weinberg equation really describes the genetic makeup expected for a population that is not evolving at a specific locus, at a specific gene. So if the data observed for the population differ from the expected values, then the population may or may not be evolving in that locus. It remains to be tested. A population in which the mating is random and none of the mechanisms of evolution are acting is said to be in a Hardy-Weinberg equilibrium. Okay, and it's called an equilibrium because in equilibrium, the population is as follows: all the alleles and all the genotype frequencies for that given gene, for that given locus, remain constant from one generation to the next. Okay, so if the population is at Hardy-Weinberg equilibrium, the allele frequencies and the genotype frequencies will not change, they remain constant. The Mendelian inheritance patterns preserve the genetic variation in the population, but the frequencies, the allele frequencies and the genotype frequencies will remain constant for that one gene, for that one locus, if the population is in Hardy-Weinberg equilibrium. Now we're going to talk more about Hardy-Weinberg equilibrium in the next lecture, and now we're going to stop here. And I will see you soon.